Action.
Okay, right, moving on. Right, so I did have an amazing lesson planned for you. Now you can't have it. Hold on, I'm trying to. No, didn't.
How come we don't run the face shields?
No, let me explain about face shields. Okay? So, the stuff is wavy. So you get seasick all the time. Because every time you move your head, you go... And it gives you a headache, yeah.
Are you sure that's the mask for the children?
Or a combination of it. No, it's the mask. Okay, I'm missing a whole lot of kids, aren't I?
Vishani? Shanae?
No, that's fine, but like, what's this?
This is your class.
No, but then these kids expect me to do all online I love you things. No.
Now we can do online too.
You can do?
What?
How are you going to do online with this stupid computer at 19%?
We can just wait until it's done. 90%, that's not bad. We can do this.
Okay. Don't scream at these kids. What's the fun? Okay. Alright, so let's just quickly go back and remind you what a synapse is. Okay, can anybody see my pens? Where are my pens?
Your hand?
On the board,
There,
No,
that's in the
no.
packet. In the packet.
No, those are different ones. Because they're too thin and they write too light. But we'll try this because I have lost all the other ones. Okay, right, so go ahead. Okay, so remember that the explanation, the description of a synapse is, it is a region where the synaptic knob of one neuron lies close to but does not touch the dendrites of the following neuron or a cell of an effector organ. So it could be a muscle cell or a gland cell. But remember, the important thing is that the synaptic knobs do not touch. So, if this is the presynaptic neuron, and that is the synaptic knob, the important structural things about the synaptic knob is that it has vesicles which contain neurotransmitter chemicals. In addition to that, it contains renin mitochondria.
No, I didn't think it
Hmm?
I didn't think it in the camera.
Oh, no, I'm not in the camera, I'm talking to Jessie.
Oh, I was in the camera. I just like
Can you see the mitochondria?
Yeah.
Can you see them?
I can see that. Not
Okay.
clearly, but I can definitely see that.
Okay, all right, everybody happy with that? And then remember that the postsynaptic neuron, the dendrite would lie close to but not touch the presynaptic neuron. And so this little gap here is called the synaptic cleft or the synaptic gap. And remember that that is filled with fluid. Okay, remember that on the membrane of the postsynaptic neuron are tiny little channels, and each of those channels under normal circumstances is closed, and there is at one part of that channel a little protein receptor Okay, and that's key in opening that channel. So remember that the channel is closed under normal circumstances. Okay, but the channel has a little
Sorry, ma'am.
a little protein. In other words, it's got a special shape because it's a protein, and you know that proteins are able to form specific shapes. A little protein receptor at one part of it, and if a neurotransmitter attaches to that protein receptor, then the channel opens. And when the channel opens, ions are able to flow through, and when ions flow through, it starts an impulse in the postsynaptic neuron. Okay, you happy with that? So each of these little channel things has got one little spot to which the receptor, the neurotransmitter is able to attach, and that causes the channel to open. Okay, you happy with that? So remember that the way in which this thing works is an electrical impulse, 53, an electrical impulse will move through the neuron that is pre-synapse. When it reaches the synaptic knob, some of those little vesicles move to the surface, and they are then able to fuse with the surface membrane. So if this is the surface membrane, and there's a vesicle containing neurotransmitter molecules, it is able to fuse to that membrane and therefore open up, and the neurotransmitter molecules are released into the synaptic cleft. Okay, you happy? All right. At that point, the neurotransmitter molecules then diffuse through the liquid across the synaptic gap, and if they do, then they are able to join to the little receptor molecules of the channel proteins and are able to open up those channels. When the channels are open, ions that are present in the synaptic fluid are able to flow in to the postsynaptic neuron. When those ions flow in, that starts the electrical impulse carrying on in that postsynaptic neuron. Okay. But at that point, those channels are then open because there's a neurotransmitter attached to the receptor. So the channels are open. It's terribly important that they close again. So to remove that neurotransmitter from where it is attached there is an enzyme present in the synaptic fluid. And that enzyme is able to break down that neurotransmitter so that it goes back to being its little component molecules again. And as soon as it's broken down, it can't attach to the channel receptors anymore. And so the channel receptors then close. Those little components diffuse back across the synaptic gap. When they reach the presynaptic membrane, they're taken up again. They are reassembled back into being little neurotransmitter molecules, and they are then put back into vesicles, and they're ready to be used again and again. The process, and this is critical, the process of taking up the components, reassembling them again, and making new vesicles requires an incredible amount of energy. And that is why there are lots of mitochondria there. So don't get it wrong, because students always think that the whole process requires lots of energy. The vesicles fusing with the membrane and the neurotransmitter being released and diffusing and attaching and the channels opening and whatever, yes, energy is required for that, but it's not a lot of energy. The fact that there are many, many, many mitochondria there fulfills the requirement for taking up the components of the broken down neurotransmitter, reassembling those little parts back into being neurotransmitter molecules, and then incorporating them into vesicles so that they can be reused. That is a high energy requiring process. Okay, everybody all right? Okay, okay. All right. Let me just go through what I expect you to do in terms of what you need to know. It's here, there. Okay. So, by tomorrow, Monday, you must have gone up on page 7 up until that heading, the central nervous system. Now, if you look on page 6 and you look at activity 6.5 on page 6, I'm listening.
Ma'am, when you say we must go up to page 7, do you mean learn it and do it? Ma'am! Do you mean learn it and do the exercises? Like all of them? And know everything? Okay.
What?
That's all I needed.
Like
No, the Gr11 wasn't a due.
Like you have to study it.
You don't have to.
No,
At no stage of the game is anybody forcing you to do stuff that you are too lazy to do.
I'm gonna have a test on that. We just want us to learn it.
Now, I'm sure we're never going to have a test on this
No,
one.
now I feel like next week.
Okay, Emily, just let me finish this while the going's good, because first of all, I'm busy growling at this, and secondly, I'm busy listening to Isabella whining, and thirdly, I'm halfway through saying something. Okay, so on page 6, activity 6.5, you see it says use the diagram in figure 6.9b. Just got to wait for Isabella to find it.
Why me, ma'am? You can carry on.
Because everybody else has found it already.
Oh, you can carry on, ma'am. It's not like you ever care about who's up to speed or not speed.
I do with some kids.
Okay, you can start.
Okay. It says use the diagram in figure 6.9b. Now, you don't have figure 6.9b, or you don't in these notes, but I have scanned it and I've put it into the general folder for the nervous system on Google Classroom. Okay, so you can find figure 6.9b there. Okay, so ideally I would like you to have learnt all this work.
Like learn, so like you're gonna say like what's a
And then, in addition to that, I would like you to, if the mood moves, to do all the questions and the activities.
When are we going to make them?
You're going to mark them when the memos are released on Monday.
In class?
I don't think so.
Wait, I'm trying to suppress the yell. No. You kids? No.
But like, man, you know sometimes you say learn the work or what, like we're gonna have a test.
Oh, so next Thursday you'll probably have that. Sorry, my babe. Population and no.
What?
Photosynthesis and cell respiration test, probably next Thursday. Yes, my baby.
I was going to ask that because I was getting confused. I didn't know if we had to learn this now.
No, you have to learn that and the other.
So you know how you always say, man, you know how I always say learn the work, but this is what you say, learn the
I'm
work.
so defeated.
Right now.
It's like,
Which means You don't actually have to learn it, you don't actually have to
Okay, Bless is now ducking, so that when I throw something at you, it's not going to hit her. It'll probably hit Tony. No, now this thing's doing the circle of death. It's not even saying scanning, it's just doing the circle of death.
Who else wants to go home and cry for the amount of work we have next week? Oh, me. Me too, man. It's so much.
You have no idea. You haven't, since lockdown started, 7 days a week, I have worked a minimum of 12 hours a day. Because we had this has made such an extra amount of work for us. It's incredible. I mean, like when the kids do an assessment, there's 6 kids that are away that have to do an online assessment. Now I've got to worry about the online assessment. How am I going to do the online assessment? I've got to print all the things out, then I've got to mark it, then I've got to scan it, then I've got to send it back. It all just adds a huge amount. This thing just adds hugely.
How are they going to do the test if they're not at school on Thursday? Because then can't they just cheat?
So we set up a Google Meet, and the things on them, we watch them while they write the test. And
I still think it's pretty easy to cheat that way around. Very.
Just stick a piece of paper there behind the
Sydney?
computer.
so now I know that you're the cheater of the world.
No, I don't cheat. Like, okay, I have cheated.
Don't tell me that. I want to believe that you're a nice child.
But ma'am, I mean, like, if you're sitting in front of a computer and you could literally have your book like right next to you.
No, but we watch that.
You can have it stuck on the computer.
So the first thing is the more you look there, the more you're not going to finish. Because you're under huge time pressure.
How much time are we allowed?
But there's always, I mean, biology tests and things are always like really, okay, let me put this password in while the date is good.
And is the biology test next week Thursday? Does it include like a practical or is it just theory and
No,
calculation?
it's just theory and calculations. It might include a practical component, like if this was the investigation, what's the dependent variable, what's the independent variable, what were fixed
Probably
variables.
like, draw up a method and you're sitting there like, oh.
Magnification. Yeah, I mean, we usually do.
Yes, always. You see, listen to you. In addition to this, we also have to put up with you whining.
You missed us whining.
Oh my
You did.
Oh.
No, I didn't miss you whining. I missed being able to talk to you and making you whine. No.
And then saying, wait. No, I don't.
Okay. Let's see if we can get this to work now. Everybody 100% happy about that. So the thing about learning, when I say learn something, if you don't learn something when I've taught it to you, and next lesson builds on that, and the expectation is that you know what I'm talking about. If you didn't go through that work and you don't know what I'm talking about, when you have to learn for a test, it's going to be a nightmare. Because each lesson is like, it would be like you going into maths without doing your maths homework. You just wouldn't do it.
Sometimes I feel like if you learn the work now and you don't have a test, it's just wasting time.
No, it actually isn't. It goes into your long-term memory if you do it like that. And it means that when you learn for a test, 70% of it's already in your long-term memory. It takes you a lot less time. I've done a lot of research into this. I've read a lot of research that other people have done into this. I promise you it works.
Then you kind of like sit there and you're like, did it start with a J or a G? And then you can't get it and you just start to just put a K there for the sake of putting a K.
So he talks about spelling or like.
Yes! I like words. You're supposed to be like, it's like,
Why would you put a K? Why would you just put a G or a J?
because we don't know what's going on.
So then you put a U or J. If you've got a slight thing.
Sometimes I even forget to eat, and I was like, where did that come from? It's so difficult.
Tony, you need a good stress management.
Maybe it's stressful.
Tony, you're prepared for real life.
No.
No.
Stress and anger.
I thought
Life
I
is
was
a stress management. Come on.
No, both.
Just don't do it.
Right.
I feel like we should be learning about taxes.
A scholarship.
Well, I mean, if you're really bad, you should get a scholarship.
You don't get scholarships
I'm
for
really
rehab
bad.
centers.
And you know that because you've tried.
Yes, around 100 times.
scholarship for
Okay,
real.
there was
Yeah.
a sociopath school.
Yes,
Isn't
that
that
was
just called a
definitely that one.
sponsorship?
Huh?
Isn't that just called a sponsorship?
No.
Tiny.
Is it like the sponsorship?
Tony, if you ended up in a rehab centre, your mother would just push you through the gate and lock the gate and fine. See you, Tony.
No, you don't see that I'm mental.
Okay.
The bit is not going well.
It says moose party.
It didn't. It said mouse party.
Oh,
Not only that, she can't breathe. Okay.
It killed it.
All right.
Man, that puzzle was super fun, you know.
Mom, can I tell you a funny story about Tony?
No,
Why?
because I'm going. Okay. Trouble is, it's going to start the mouse party if I do this.
the moose
Mouse
party.
party.
Every time she's mouse or man, it's not the same.
No. Why isn't it doing an audio? Oh, God, I hate school.
Now, why are you a teacher? General question, you don't like kids, you don't like school, you don't like
Why?
work. Like, why are you a teacher?
Because
Aren't
I
you a
love
biologist or something?
it. No, I love it.
Oh, yes, sir.
Okay, I have no idea why this thing's not making a noise.
It's fine, just play it, man.
Man,
No.
why do people so crazy?
Where's the speaker, man?
Yeah. Wait, maybe this one.
Man, I can hear your voice over.
The synaptic vesicles to release
That's
acetylcholine,
starting
a
again.
neurotransmitter molecule.
Looks like Sarah.
Okay, shut up, mouse party.
House party.
Okay. All right, going. Wait, start. Oh, why is it not?
Put it down, man.
Oh,
Calcium-ion channels to open.
okay, trying again.
Action potentials arriving at the presynaptic terminal cause voltage-gated calcium-ion channels to open. Calcium ions
So
diffuse
that's a
into
little
the
bit
cell
detailed,
and
you don't
cause
need to
synaptic
know that.
vesicles to
But you
release
know from
acetylcholine,
here.
a neurotransmitter molecule. Neurotransmitter molecules, acetylcholine, diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium channels. This causes the ligand-gated sodium ion channels to open, and sodium diffuses into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced.
Okay, do you get it?
It looks like celery. With green things.
Okay, right. Did you understand what it was saying? So there was a little bit too much detail at the beginning, and it called the channels ligand channels. You don't have to know that. Yeah, Matt.
Is the lutein the synaptic knob or the synaptic fluid?
So this is the synaptic gap or the synaptic fluid. So let me say it again.
Action
Synaptic
potentials
knob.
arriving at the presynaptic terminal cause
Okay.
voltage-gated calcium ion channels to open.
So know from
ions
here
diffuse into
on
the cell
what
and
happens.
cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Neurotransmitter molecules, acetylcholine, diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium channels. This causes the ligand-gated sodium ion channels to open, and sodium diffuses into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced.
Okay, all right, so then it's saying new impulse in the postsynaptic neuron. Yes, Erin?
Ma'am, does the enzyme in the fluid not try to dissolve the neurotransmitter when they're diffusing?
No.
Only
No.
when they...
Yeah. That's hugely complicated. I have no idea. So we're going with no.
Thanks.
Okay. All right. This made sense at the time. I'm not sure why. Okay. Now, okay.
The reward pathway in the brain is a complex network of millions of nerve cells. Signals travel along the reward pathway from one region of the brain to another. Nerve cells communicate with each other at a junction called the synapse. Here, signals move from one neuron to another. The small space between the two cells, called the synaptic cleft, is where the action takes place. The cell that will send the message is loaded with sacs, called vesicles, that are filled with neurotransmitter molecules. There are many types of neurotransmitters. The type used along the reward pathway is dopamine. The cell that will receive the message is coated with dopamine receptors. An electrical impulse triggers vesicles in the sending cell to dump their contents into the synaptic cleft. The dopamine molecules attached to the receptors on the receiving cell. Dopamine and its receptor are specifically shaped to fit like a lock and key. When dopamine is docked, the receptor triggers the production of a second messenger molecule. Dopamine is released from the receptor and it goes back into the sending cell through reuptake transporters. Here, it will either be packaged for reuse or broken down. The second messenger can interact with other molecules to produce effects within the cell. It can also trigger an electrical impulse which travels down the axon. Our receiving cell is now a sending cell, and the process repeats. The impulse reaches a synapse at the axon terminal, stimulating neurotransmitter vesicles to dump their contents.
Let's see, it's a different
So, when does an
neurotransmitter,
impulse
different
stop?
shape.
First, if the signal is weak and too few neurotransmitters bind to receptors, the receiving neuron won't fire an impulse. Second, certain types of neurotransmitters are inhibitory, like the ones shown here. Inhibitory neurotransmitters stop nerve impulses. The neurons presented here are just a small part of a vast system. In elaborate networks, neurons in the brain work together to control sophisticated functions, such as learning, mood, and memory. In these networks, one neuron may connect with up to 1,000 others.
Okay, all right. So basically, this is like a generalized version. There are slightly different versions depending on what the neurotransmitter is. So in some cases, the neurotransmitter doesn't open a channel, it just causes changes in the membrane. And that was what this one showed you. Just learn it as I've taught you. Okay, don't obsess about it. Okay, so different neurotransmitters in different neurons, and they will always have a particular receptor on the other side, on the postsynaptic membrane. Okay, now, certain drugs act